Right. Okay, so this is the second video in what will probably be a multi-series set of videos dealing with specific research that I'm doing in the field of comparative psychology. So that is to say the study of animal cognition, so looking at interspecies differences in the structure of cognition. So in this case, I'm going to be talking about a paper which I published about roughly 1 year ago, which is on a thing called the general insight factor. And this is a measure that relates specifically to different types of physical cognition measures in parrots. So I will all be made clear as we proceed through this. So this is the paper, and the paper specifically looked at two things. It looked at replicating string pulling using a parrot species called the Greater Vasa parrot, or Coracopsis vasa, which is its scientific name, in order to see if it sort of had this capacity that other researchers think that it has. And we also wanted to look for this general insight factor using data not just on vasas, but data on other parrot species as well, which we could use as the basis for comparison. So this research was done in conjunction with Peñaherrera-Aguirre, who you'll see quite a lot on my comparative cognition research. He's my primary collaborator on this topic, and my brother Anthony, who actually did a lot of really nice editing, putting together the ethological data into a nicely sort of presented way, which you can download from the supplementary material associated with this paper. So that's the authorship. So what is insight-based problem solving? Well, it's any kind of problem that needs to be solved through having to sort of imagine a potential solution to the problem before implementing the solution to that problem. So the classic example of this is known as the string pulling paradigm or string pulling problem. And it and variations on it, because there are very complex variations on this basic string pulling problem, have been used for centuries, in fact, to probe insight-based problem solving in birds in particular, although it's also used in chimpanzees and other mammals. Now, Pliny the Elder was in fact the first person ever to have recorded an animal being able to solve this kind of string pulling problem. And he looked specifically at captive goldfinches, which were kept in a state of dehydration. And in order to retrieve water, they had to pull up little buckets containing water in order to get at the water. You can see an example of this on the right, although I believe in this case the goldfinches are retrieving a treat or a food item from the buckets. I don't think they are retrieving water from the buckets. But this sort of trick was something you could do quite regularly, and it was something that was known even as far back as the time of Pliny the Elder. Now, it's a much more complex problem than people think, because it requires you to be able to coordinate these body parts in alignment with a sort of means-ends-based reasoning approach. So the idea is, like I said earlier, you have to imagine a solution to this, and then you have to work out how to coordinate your body parts to actually realize the solution to this. So there's debate about just how cognitively demanding string pulling really is, and some have argued that, in fact, the ability for an animal to pull strings and solve problems through pulling strings might actually just be a sort of byproduct of having various functionally specialized feeding adaptations. So, for example, having a foot and a beak might be and being able to use these things to pull things out of the ground, such as worms, might be something that sort of biases you as a species towards being able to solve this problem spontaneously without necessarily having to engage in any higher level cognition. So one way around this is to actually come up with more elaborate forms of string pulling. So this would include things like having a dummy object on one string and having the actual object on the other string, and then working out if the animal can work out which one contains the actual reward and which one contains the fake reward. Or another version of this would be to have one string which is connected to the reward and another string which is connected to nothing. And the animal has to work out causation, essentially. If it pulls on one, it gets nothing. If it pulls on the other, it gets something. Or perhaps where you color code the strings, so it realizes that only pulling the red string gets it the reward, pulling the blue string gets it a dummy. So there's lots of variations of this paradigm, which are much more complex than the one you see depicted there on the right. Now, based on Ostaf's review, there was a big review which was published about 15 years ago. No, published about 7 years ago, rather. He found that this string pulling capacity, especially in its most simple form, which is where you just have a single string and one reward, essentially, the animal has to reel it in, is quite common among parrots. Many parrot species have been observed being able to solve this problem, but also other birds have been observed to be able to solve this problem as well. So it's not just parrots, but as you might expect, corvids as well, which are known, these are crows, ravens, jays, magpies and their allies, which are known to be very intelligent. They can also solve this problem. Bullfinches, as we've seen, goldfinches, which are the ones depicted there on the right. The smaller passerines can solve it, and grackles as well. These are brood parasitic passerines in the the Icteridae family. Include things like the genus Quiscalis. You might know them as cowbirds. They're quite common in the States, particularly in the southeast. So basically, many birds can solve this problem, and as I mentioned, chimpanzees can solve it, and other primates can solve it as well. And Audit referred to it as a sort of a select club, because even though there are many species that can pull strings to solve problems, compared to the total panoply of species out there, it's still a minority. So there seems to be something special about those species that are able to solve this problem versus those that apparently cannot. Now, the first part of the paper was, and really this is what the paper started off life as, essentially, as a replication of basic string pulling in this particular species, Coracopsis vasa. And I happen to have one as a pet. But I also wanted to establish longitudinal stability, because I had actually recorded 2 sessions of the bird pulling strings, one in 2013, and I recorded a second session in June of 2020. And I was able to not only show that the bird could solve the string pulling problem, and the above 4 frames there you can see are from the 2013 trial, and this is the bird simply just reeling in the string, and it's using looping, and it's using tugging on loops and all sorts of stylized response processes in order to reel up the bit of cheese, which is the reward he has there tied to the end of that string. This particular bird is very fond of cheese. So yes, he was able to do it really without any training, without any prior exposure to the task. And in the 2020 session, we refamiliarized himself with we allowed him to refamiliarize himself with the task before giving him an actual go. We were able to calculate the amount of time it took for the bird to solve the problem, and we were able to calculate an efficiency by basically looking at the ratio of string pulling events which led to the reward being pulled closer to the bird versus those that were led to the reward either slipping away or not. These are instances, like for example in that bottom right-hand frame you can see there where the bird is pulling on the loop. It's not actually pulling the reward closer to it. So that was an example of a miss. So that tells you that out of all the beak string contact events, what proportion of them led to a successful sort of pulling of the reward closer to the bird. Now, there was a difference, and it seems that in the more recent trial, the Vasa was slightly less efficient, 43% efficient in fact, versus 67% efficient. However, that difference was not statistically significant given the small number of observations. So we had to conclude on this basis that there really was no significant longitudinal change in the bird's capacity to pull strings. And the other thing we noticed was that we weren't the first people, though I might have been the first person to ever have conducted an experiment on string pulling in the Greater Vasa. I was not the first person to have presented data on this. That honour belongs to Peñaherrera-Aguirre, who wrote a PhD on string pulling in various parrot species back in 2014, and she actually examined Vasa parrots at the I think it was a Skegness Zoo. She looked at 9 birds, whereas we looked at a single bird, she looked at 9 of them, and she was able to get them to solve not just this type of string pulling problem, but also to solve more varied kinds of string pulling problem. And her data are very useful to us because they allow us to do comparisons between Vasas and other parrot species, evaluated using the same measure. But our analysis was different subtly in the sense we just used a single bird. So there was really no opportunity for distractions and no sort of collective problem solving component. This is literally one bird, one string, and the data on that basis can support certain other sort of theories about what goes into solving this problem. In other words, the bird doesn't need to be around other birds to solve this problem. This species can solve it on a singleton basis, which is a useful data point. So we've kind of made an additional observation there that's additive, I think, with respect to the insights gained from the Peñaherrera-Aguirre thesis. So that was part one, and really originally that was all the paper was. It was just that. So we decided to sort of beef it up once we managed to get hold of the data from Peñaherrera-Aguirre's doctoral dissertation. She published it all in there, so it's all available. And we thought we'd reanalyze some of her data because she did some really outstanding analyses, but we saw some ways in which these data could be analyzed that might sort of support more fundamental insights, or might allow us to test more fundamental theories about what might be going on at the species differences level in terms of string pulling across different tasks with different levels of complexity. So one of the questions is there a general factor, like a big G factor, among species differences in performance across these 5 tasks? We had 14 citizens, that's parrot species in total. They were evaluated using directedness, color, continuity, connectivity, and inhibition. And these are just different configurations of string pulling which test these different faculties, like the ability for the animal to be directed to the correct solution versus the ability for the animal to choose the right color, the ability for the animal to work out that there's continuity between a particular string and a particular reward, connectivity and inhibition, which is like a sort of a delay of an impulse control measure. So essentially, there's all these weird and wonderful ways in which these animals can be evaluated using variants on string pulling. And Peñaherrera-Aguirre presents data for 14 species. So the first thing we did is we examined these data to see if there was any evidence of phylogenetic signals. And the reason this is important is because where you have phylogenetic signals, you have possible autocorrelation effects in the data that mean you have to use different kinds of software to compute relevant regression parameters. So we wanted to see if there was any potential phylogenetic issues with these data that might mitigate the use of simple analyses. And we also managed to collect, again from the dissertation, this is based on data that was presented in the dissertation, data on covariates such as brain size of the animals, or relative brain size, and fission-fusion flocking intensity. And that's essentially a measure of the degree to which the animals are integrated into flocks whose composition varies over time in response to different sort of environmental challenges. So it's kind of flexible flocking where animals opt in and opt out of the flocking, and it can affect the size of the flocking, and that this can occur in response to all sorts of different things. This fission fusion flocking is thought to be a major catalyst of avian evolution, particularly avian cognitive evolution. And because it seems to be, Peñaherrera-Aguirre found indications that using a different regression design to the one that we used, that fission fusion flocking was a fairly robust predictor of different kinds of string pulling ability across the species. And one implication of this is it might be a source of general covariation among these different measures as well. It might be something, an evolutionary history that's essentially causing these things to collapse into a general factor rather than just being sort of idiosyncratic to each measure. And we also computed mean pairwise difference because we wanted to see if that was in any way moderated by the loading of any general insight factor onto these indicators. So we'll get onto more about how we dealt with those later. So this is the result of a phylogenetic signal analysis, and basically what we found was, when computing a thing called Pagel's lambda, which measures essentially the correlation between branch length in a phylogeny and the difference between species means for any given trait. The stronger that correlation, the more phylogenetic signal is present in the data. So what you really want are low values, because low values indicate there is no phylogenetic signal in the data, and it means that special precautions for phylogenetic autocorrelation effects are not needed. So what we found was that across these indicators there were no statistically significant phylogenetic signals. The one which came closest was connectivity. So in the case of connectivity, the effect size there suggests that there is possibly a tendency toward branch length and node distance being correlated with each other in a way that's consistent with sort of phylogenetically conserved trait and possible phylogenetic autocorrelation. But this effect did not actually reach conventional significance. It was slightly under a two-tailed p-value of 0.05. So strictly speaking, none of the measures had any significant phylogenetic signal. So on that basis, we were able to proceed with the next analysis. But one thing I will say is that the results of phylogenetic signal analysis are generally not stable, or not too stable, when the n of species is below 20. So the only reason we really did this was because it's sort of obligatory in these kinds of analyses. And it kind of hints at what we might find if the sample size was larger. I don't think we'd find any phylogenetic signal if n was bigger than 20, except possibly in the case of continuity. I suspect that would probably become significant. Anyway, moving on. We will assume there's no problematic phylogenetic autocorrelation effects in these data. So on that basis, we computed a suite of parameters. The first one, which is the most significant here, is lambda. Now, this is not the same as Pagel's lambda. This is a different lambda. This is a lambda that's used to refer to the degree to which a latent variable loads onto its constituent indicators. So in this case, we have our four different types of string pulling: directness, color, continuity, connectivity, and inhibition. And we have the loading of the general factor among these onto each of its constituent components. And these estimates are produced using unit-weighted estimation. And the way this works is you simply average the standardized scores for your indicators, and the average becomes the general factor or latent variable. And then you do part-hole correlations between each component and the average, and that gives you your factor loadings. And if you square those and average them, you get the proportion of variance accounted for by that latent variable. And if you take the sum of the squares of those loadings, you actually get the eigenvalue. So you can compute all of those relevant factor analytic parameters using this very simple technique. The reason we used unit-weighted estimation is because it's recommended when case and or variable number is low, as in the present study. Because it tends to counteract sampling error through some complex mathematics, which I won't go into. But it counteracts sampling error in a way that leads to the resultant factors being more highly generalizable. So this is one of the advantages of a technique. And you can see that each of these loadings are statistically significant, but unambiguously large magnitudinal cases. This suggests that there really does want to be a substantial general factor there. We also computed species differences in performance as a function of fission fusion flocking intensity. So we just took the degree of fission fusion flocking intensity, I think it was scaled from 1 to 3, and we just correlated that with each of these measures. And then we did the same thing with relative brain size. So that's the species difference correlated with each indicator. And then we did the same thing. We did another thing where we took the average pairwise species difference for each pair of species, and then just averaged the D, the difference between each pair of species. So we have 14 species, and that's quite a few pairwise comparisons. And we did that for each ability, taking into account the different numbers of species available for each comparison. So these parameters are used for the moderator analysis. So let's first of all focus on what the taxonomic differences look like with respect to these measures. Well, with the general insight factor, the unit-weighted general insight factor, which is what we're calling it, the general factor among all of these different string-pulling modalities, when we break the sample out into its respective families, we have three cytosine families represented here among the 14 species. We find there are differences, but none of these differences are statistically significant. And again, that's because we have relatively poor sample. We have small numbers of species per family, essentially. But there do seem to be some differences here, nonetheless, which you can see in this box plot. Now, this is the more interesting analysis. We used an ancestral character reconstruction, which is based on fitting the means to a phylogeny, essentially, and then you can use a heat map, essentially, which is what that is. It goes from blue for cold, meaning very low association, red to hot, meaning very high association. Everything else is green if it's sort of middling. So you can use this to identify sort of where the hot spots are in the data, or the cold spots. So in the case of vasas, you see them there, Coracopsis vasa. They're one of only two that have a red, and the vasa belongs to the Psittaciulidae family. They're one of only two species that's red, right, that actually has a very hot, it's a hot point. It's a high signal point in the heat map. So that means they have a very high level of general insight capability relative to other species. The only other species of parrot that comes close is the Phorpus conspicillatus. It's not one I know. It's a Phorpus parakeet, a spectacled parakeet, a parrotlet rather, which is a very small type of parrot. They also seem to be quite good. But of all of the species, the Greater Vasa is actually the one with the highest general insight factor ability. So it beats all of these other parrots in terms of general insight factor. Now, I told you we'd return to the moderation analysis. So these are simple analyses using basically a vector correlation method. So what we do is we take the correlation, which I've already showed you for each of these indicators between, say, brain size, relative brain size rather, and each species score on each indicator, and then we correlate that vector across subtests with the vector of G loadings, or rather GIF loadings. We do the same thing for fission fusion flocking intensity. That's just the correlation between that and insight for each species, and we then correlate the vector of that with the general insight factor vector. So this is standard sort of MCV, or method of correlated vectors. And we finally did it for the pairwise comparisons, again, plotting those out as a function of G loading for each, or GIF loading, I do apologize, for each of the measures. Now, the vector correlations are not too great. It's a sort of middling magnitude one for brain volume. For fission fusion flocking, it's very high, it's 0.831. It's very high magnitude correlation. And for the mean pairwise differences, again, it's sort of middling. So the only one which seems to want to do anything here is fission fusion flocking. Now, I'd say that the caveat here is because we only have 5 scales. None of these effect sizes are statistically significant. So if we look at fission fusion flocking, that is going to be the most interesting one, simply because it is the closest to being statistically significant given conventional cutoffs and available power, and it is also the one which we're most interested in, because it's the one that we theorize might actually be driving the covariation among these abilities, which is sort of one of the main reasons for wanting to test for these moderation associations. So of all of them, fission fusion flocking intensity seems to be driving correlations among abilities, not just the level of the ability, but also the covariation among the abilities, which is interesting, and this is something that could perhaps be revisited in future comparative psychological research, because for birds at least, fission fusion flocking seems to be a very significant factor in their the evolution of their cognition. And this is consistent with a great deal of literature on avian cognition, which has been produced thus far. So I'd just say this is yet another indicator. So in conclusion, to sort of wrap this up, simple string pulling can be replicated in a singleton context using an adult male Greater Vasa parrot. And this adds data to the available data on this taxon, because we can show it can solve this problem spontaneously without there having to be other vases around. So there's no collective problem solving, there's no collective component to its capacity to solve this problem. It seems that the bird can just go ahead and solve the problem. No longitudinal age-related decline. There is in absolute terms, but it's not statistically significant. So maybe we follow up in another 7 years and see what happens. Clear indications of a general insight factor among the 14 species, inclusive of the Greater Vasa. This is based on our reanalysis of Krasheninakova's data. There's variation among species and also among individuals, but among species more relevantly. And the Greater Vasa actually scores the highest on this of all of the other parrots. And this between species covariation is directly analogous to what has been termed the big G factor, which is found among different measures of cognition in species level data, often involving 60, 70 different species of primate. This is a data set that my colleagues and I have published on quite extensively. The first finding of this was Reader et al. 2011. So there seems to be some parallels here, but the question is really, is this GIF factor, this general insight factor, the same thing as the big G factor? And is there really a big G factor in birds? Well, we sort of hypothesize on a hierarchical factor structure of species differences, which might look a little something like this. So these are just two of several different domains that could be subsumed into stratum 2. So we have a general insight factor. We have our general insight factor there, and we've also put that as being co-equal to a general tool use factor. And of course, there could be many other factors populating stratum 2, but the reason we chose to represent general tool use there as another stratum 2 factor is because in the case of vases in particular, they seem to be able to use tools, which is interesting. So apparently they're not just excellent string pullers, but they're also able to use tools spontaneously, which very, very few animals are capable of doing. And in the case of vases, the tools are quite complex. They involve the use of shells and stones, which they sort of grind together in their beaks, almost like a mortar and pestle, in order to grind up seeds and scrape calcium and all that sort of thing. And the other thing they do is share tools among themselves, which I think might be unique to vases. I don't think anyone's ever observed that in other Cytosine tool users. So vases seem to be high on both general tool use, if we take those two stratum 1 manifest indicators as good proxies for what could go into a hypothetical general tool use measure, and they seem to be high on the general insight factor as well. So at least in that one species, the sort of co-occurrence of these two cognitive faculties hints at the existence of a big G that subsumes both of these domains that would be more analogous to the one we find across different primate species. So on that basis, I'd like to say thank you for listening, and that's everything. So until